Okay, perfect. Thank you. Okay, I'm going to share my screen one more time. Please let me know if you can see my screen. Are you able to see my screen?
Yes.
Okay, perfect. Okay, I will pause every now and then so you can start asking questions. Are you comfortable with discussion while I'm presenting? Are you comfortable to answer some questions during the presentation?
Yes, we are.
Okay, perfect. Yeah, because, you know, ideally, the presentation should not take more than 40 minutes, but practically, you cannot listen to anybody on earth for more than 20 minutes. Okay, so I'm going to make some breaks to make sure that you are still following, and if you have any questions, then I can take it from there. Okay, so I'm not sure if we met before. My name is Yasmine Abusamra. I'm a professor of biochemistry and medical genetics here at Sonoran University in Arizona. Also, the Department Chair of Basic Medical Sciences and Division Director of Human Sciences at Sonoran University. I graduated from Khartoum University or University of Khartoum, Faculty of Science. I got my master's there, and then I continued my education in Michigan State University, where I received my PhD in biochemistry and molecular biology. I taught in Ibn Sina for about 12 years, and right now I'm with Sonoran University for about 6 years. Okay, so today your topic is RNA synthesis, processing, and modification. So can you tell me anything that you learned before today about RNA? What do you know about RNA?
That it is single-stranded.
It's single-stranded, exactly. What's the component? What's the material that makes your RNA?
Nucleotides, and it has uracil instead of thymine as a nitrogenous base.
Okay, perfect. So we know that RNA is a very important molecule that we need it for so many purposes, and it's made from nucleotides, of course. It's single nucleotide, a single-stranded molecule when we compare it to the DNA. Perfect. So it looks like you already have some information about the DNA, right? Did you take any lecture before about DNA?
We took DNA replication today.
Okay, great. Okay, so then I'm going to go very quickly on nucleic acid structures, because if you don't know the structure, you will not know the function. And even for you as future physicians, you need to understand that structure is always the key for any kind of functions. Okay, so for example, if somebody is suffering from anemia or anything, rather than to try to treat the anemia, first of all, the doctors will try somehow to make sure that their red blood cell structure is normal, okay? Because if any cell does not have a correct structure, they cannot perform the correct function. So that's why sometimes, when we start with the medical students and we start talking about structure, they don't digest it, they don't like it, because they just want to jump to the next step. How am I going to benefit from this as a physician? So yes, you have to know the structure. Everything is based on the structure. Okay, so nucleic acid structure, as we just talked about it, it contains nucleotides. Okay, then the nucleotides here, that they are formed from a base, a sugar, and a few phosphates. Okay, it could be 1, it could be 2, it could be 3. Okay, but still, as long as it has a phosphate molecule, now it's considered as a nucleotide. Okay, so on the right hand side here, I have purines and pyrimidines, and those are the bases. Okay, so this is a complete structure. Okay, and a general formula of nucleotides. Okay, now what's the difference? Sugar? No, the sugar is always the same. It's always ribose or deoxyribose sugar, but it's always the 5-carbon sugar. Phosphates are always the same, whether we have it 1 or 3, it's the same phosphate molecule. So the only thing that changes here is the base. For the bases, we have either purines or pyrimidines, and as somebody mentioned here, we have differences between DNA and RNA, and the only difference is within the pyrimidine bases. Okay, so adenine and guanine, we have them in both DNA and RNA. Thymine, cytosine also. Thymine is found only in DNA. Uracil is found only in RNA. So whenever you would like to determine whether these strands are RNA or DNA, just by the sequencing, just try to find whether they have thymine or uracil, and then you can find out. Now, I really like to give the big picture at the beginning of my presentations. So what could be the function of RNA? Okay, why do we need this RNA? RNAs or nucleotides. We need them to build up our DNA and RNA. Okay, so this is just one of the functions. Okay, so everybody right now, if I ask you about what could be the function of the nucleotides, the first thing that jumps to your mind is that their involvement into DNA and RNA structure. Yes, it's true, but is it the only function for those nucleotides? So the answer is no. We see nucleotides all over the place. Whenever we study any biochemical reaction or anything that's happening within our cells, you're going to see at least ATP. Okay, so what's actually ATP? ATP is adenine triphosphate. So is it a nucleotide? Yes, it is a nucleotide. Okay, so why do we need it? Why we always use ATP? What's the function of ATP? The question is for you.
Energy.
Yeah, energy. And nobody can survive with zero energy. Okay, so I believe that you already completed your metabolism in carbohydrate in biochemistry 1 and 2. So now we learned that we need energy. So our cells are doing their best to provide us with energy, and this is the form of the energy that's can be transferred through or between your cells and between your organs, and it is nucleotide. So this is a very important function for nucleotide besides the formation of DNA and RNA. Also, you are going to see some things like NAD or NADP or FAD or different molecules, SAM, for example. So these are containing nucleotides that are part of enzymes, or they can act as coenzymes. So the enzyme requires them to be activated to do their functions. Also, we see some kind of, are you familiar with cyclic AMP? Have you heard about it before, cyclic AMP or cyclic GMP? Yes, no? Or cyclic AMP, cyclic GMP. So those are molecules that are needed for signal transduction. So for example, if you have a hormone, and the hormone will bind like in the plasma membrane, okay? And the function sometimes it could be inside the nucleus. So how the signal from the plasma membrane reaches the nucleus if there is no like middleman, okay, in between. So we need some what's called as second messengers. So those second messengers are helping to deliver the signal from the hormone to somewhere inside the cell. It could be in the cytosol, it could be in the nucleus. So like a carrier, it's a messenger, an actual messenger, taking a message from the hormone and take it to somewhere. And mostly these signals are carried by phosphorylation reactions. So we have very important second messengers. We call them cyclic AMP or cyclic AMP and cyclic GMP, and those are nucleotides as well. Okay, we see them also, the nucleotides, we see them in numerous reactions. We need them to activate many molecules. So if I'm pretty much sure that you already have had the glycogen metabolism. So when you start glycogen metabolism, for example, you cannot start with free glucose. You have to have glucose activated to be built up into a glycogen. And this activation is through a nucleotide, which is UDP, uridine diphosphate. So these are general functions of nucleotides. I'm just telling you this story to make sure that, yes, we do have nucleotides in RNA and DNA, but I mean, we could utilize them into different other reactions. So there's going to be like a competition, right, between the formation of DNA, RNA, and all these other reactions or other functions. But our systems are so smart, so organized, subhanallah. They are doing the right job at the right time. So I'm just talking about under normal physiological conditions. Okay, now we're going to focus on our topic for today, structure and classes of RNA. So let's see here. We are always familiar with those three types, which are messenger RNA, ribosomal RNA, and tRNA. Do you know any other RNAs? If you know one, please unmute yourself and let me know. If you know it from somewhere else rather than my slide. Have you heard about any type of RNA other than those three? Are you familiar with those three? Messenger RNA, ribosomal RNA, tRNA?
Yes.
Okay, perfect. Okay, so those are the most common ones, and we can see them in humans, and we can see them even in bacteria. Okay, so in eukaryotic cells, besides, and also in prokaryotic cells. I'm going to mention, and I don't want you to learn all this section, but just to know that there are so many other types of RNAs, and we can synthesize them as eukaryotic, as in our cells. So all eukaryotic cells might contain those types of RNAs. Something like SN or small nuclear RNA or HN RNA or miRNA or siRNA. So those RNAs are needed for the regulation of the actual three RNAs, basically. Okay, so like for example, if you're using messenger RNA to synthesize proteins, and now you get your proteins and you don't need it anymore. So what do you do with it? Okay, so that's the question. You know that there are some, now we learn that we need those nucleotides to utilize them somewhere else. So if we use messenger RNA, translate it, and we get our proteins, now messenger RNA does not have any function anymore, and then we have to degrade it, and we can use those nucleotides to other function, to utilize them somewhere else. And we have that siRNA help with those degradation. And also, if you're done with it, you would like to inhibit it from, producing more proteins. So we have miRNA to do that function for us. So these are just examples, and I have a table that shows you all the functions for different types of RNAs, but I'm going to focus on these three, whether in eukaryotic cells or prokaryotic cells. Okay, questions, please. Do you have any question? Are you following?
Yes.
Now, I'm going to talk in general about the 3 types, and then we're going to focus on eukaryotic cells, and also we're going to talk about prokaryotic cells. So this is our messenger RNA. We can synthesize it, and by a process called transcription. And once we have it as pre-messenger RNA. Okay, this is the product of transcription. Okay, we get it, and it's called pre-messenger RNA. Why? Because this is not a functioning messenger RNA. We have to remove a small part here called introns, and then we have to add a cap to it. This cap is for protection, because if it doesn't have this cap, then the molecule that's responsible for the degradation will come and attack it and degrade it. We also, or actually the process will add poly A tail, and this tail it's highly required for translation process. So this is how we synthesize it, and this is going to be processed and getting to the final product. For ribosomal RNA, there is a nice comparison between prokaryotic cells and eukaryotic cells. Okay, so they form the ribosome. Our RNAs will form the ribosome in both cases. So the ribosome basically are RNAs and some proteins. In prokaryotic cells, we have about 55 plus proteins, and in eukaryotic cells, they are about 83 proteins. So how do you define the ribosome? Okay, you know that ribosomes are organelles in your cells. So if somebody asks you about the ribosome, tell them that ribosomes contains RNAs and proteins, and the majority of the ribosomes are RNA, and basically it's ribosomal RNA. So we have some differences, as you can see here. Their coefficient or sedimentation coefficient of the components that they are found in here in prokaryotic cells, and also they are different from the ribosomal RNAs that are found into eukaryotic cells. Okay, how about the last one, which is the tRNA? Okay, tRNA, always known as an adapter. Okay, so later on I'm going to show you what do we mean by that. So it's called an adapter because when it comes to translation, this tRNA, as you can see it will be synthesized as large script, and then it will be chopped somewhere, removing some nucleotides and also modifying some nucleotides. So if you can look at the left hand side, can you see my cursor here? I'm moving my cursor. Can you see it?
Yes. Yes.
Okay. So those are unusual nucleotides. We know that our nucleotides are A, T, G, C. So in the tRNA, you see structures like this, or these, or T's. So they are different from the normal ones, and because the tRNA plays a double role. So it bounds to amino acid, and here it has to read through nucleic acid. So those are two different languages. Okay, so amino acids, you know that they have, like, adenine, what do we say, glutamate, proline, those are the amino acids. Nucleotides here, those are the adenine, thymine, cytosine, and guanine. So these are two different languages which are, at some point, this tRNA has to make sure that reading nucleotides and having amino acids at the same time. Okay, so that's why it needs to modify some of their nucleotides to match their different functions. As you can see here, we have different loops one of them to read along the messenger RNA, and some of them just to make sure that they are attached to enzymes and having this perfect fit to continue their functions. Okay, so this is our list that shows you different RNAs with their different functions. So let me just randomly read through miRNA. So it will regulate gene expression typically by blocking translation of selective messenger RNA. So this one, if it exists and it has a target messenger RNA, it will block it, and then we cannot continue the translation process. Because why are we making messenger RNA? Why are we making ribosomal RNA? Why are we making tRNA? So we are synthesizing them basically because we need to make or to synthesize proteins, and that's the perfect goal for RNAs when we start talking about them. So after these three, if you look at any one of them, they are not involved directly in protein synthesis. They are just regulators, okay, for the existing RNAs over here. So that's why we choose to talk only about these three. Any questions so far? Are you okay with my speed? Or you want me to slow down, or you want me to repeat anything for you? Or you're good?
It's all clear.
Good.
Thank you. Okay, now we're going to talk about the actual transcription. So what do we mean by the term transcription? Transcription is RNA. So what do we need to start this transcription? We need to have a DNA and then read along it and synthesize an RNA. So this is basically what we call it the central dogma. So what's the central dogma? We have our DNA, okay? It could be replicated as we just learned, or we can just go directly to transcription to synthesize an RNA and finally proteins. Okay, so just to tell you a secret here that we are studying all this stuff just to make sure that we have the correct proteins. So why proteins are so important that we're spending so much time and putting so much energy to study all these processes? Because proteins are the working horses for our cell, okay? So if you decide for some reason that you want to block all your proteins, then the cell will die, okay? And if you heard about a process called apoptosis, which is programmed cell death, some of the cells, they are affected, okay? And your system or your body will decide to eliminate it, to get rid of that cell. The first thing that happens, the proteins will die, okay? Because without proteins, there's not going to be functions, the actual cellular functions. So proteins are so essential, okay? Of course, I'm very biased to proteins because this is the area where I did my PhD. I did it in enzymatology. But proteins are amazing. And wherever you go, wherever you study, wherever you do, you always find some sort of protein involved in there, okay? If you study carbohydrate metabolism, you need enzymes. Enzymes are proteins. If you study lipid metabolism, you need proteins, which are proteins in form of enzymes. If you study now, if you are talking about different processes, just getting the information from the DNA, even to get to these proteins, well, you also need proteins. Okay? So proteins are essential everywhere in our lives, and we have to have a continuous supply of amino acids to build up our proteins. Proteins cannot be stored in your cells. We always have an amino acid pool. Okay? So it comes certain times to build up your proteins and use them. When you're done, we have certain group of enzymes called proteases. They will degrade this protein, release the amino acids until we get to another process that requires proteins, and we start to synthesize it. So the central dogma starts from DNA to RNA to proteins. Just to remind you, what are the molecules that form DNA? Can somebody help me with an answer, please? What's the molecules that synthesize DNA? Please unmute yourself and let me know. Do you know how we can make a DNA in our cells? Okay, how do you define? Yes, nucleotides. Perfect. Thank you so much. How about RNA? We're moving from DNA to RNA. What do we have here? Which molecules?
Also nucleotides.
Exactly, also nucleotide. Now we are moving from RNA to proteins. Which molecules that are forming your protein?
Amino acids.
Amino acids.
Okay, amino acids. So now you see here nucleotides. Now we change. It's called amino acids. So that's why from here to here we just make it like a transcription. But when we change the language from RNA to proteins, we call it translation. Okay, translation ترجمة صح? Okay, so we are translating the language. So that's why when we get to the proteins, we have to translate, okay, this language from nucleotides to amino acids. And unfortunately, that we're going to stop until here to RNA, and we're not going to continue the translation. That thing should be, for the next lecture. But this is the idea, is that we need to read along the DNA certain sequence here over the DNA and make sure that that sequence has to be expressed. So the whole process called gene expression. So gene expression means that there is a gene here that go all the way through different processes to be expressed into a protein. Okay, so this is our DNA. Our DNA is huge, okay, although it fits in the nucleus of a very small cell, but still it's huge, and it has so many nucleotides in it, but only about 3% of our genome, the whole DNA, only about 3% can be expressed into proteins. So we are giving you this lecture and many more to go or to come, just to study 3% of the DNA. Only 3% are genes. And when you say a gene, how do we define a gene? A gene is a sequence of DNA that can be expressed into a protein. Okay, so now we are running after this. We want to make sure that we get to this point. And this is why all these are very essential. So if this is what we need, and we have the genetic information here in our DNA, why do we have to go through RNA? Why do we need to make this messenger RNA? We are making it because hypothetically we have what's called genetic code. The genetic code cannot read anything that contains thiamines. It doesn't read a nucleotide from a DNA. So it's always uracil. So that's why we have to make this step, make a transcript. The transcript then can be converted into a protein. So let's see how we can do that. We can do it by certain enzymes called polymerases that they are able to bind to the DNA. Okay. Of course, some regions are expressed okay? And some regions or the DNA cannot be expressed into proteins. So now I'm going to ask you another question. If only 3% of your DNA can be transcribed into proteins, and this is the main function of our DNA. It's giving you the genetic information to make your proteins. And of course, the proteins are not just for function. I mean, the color of your skin will be produced by proteins. Your IQ is related to your protein. The texture of your hair is related to your proteins. So we are trying to express those features, or we call them traits, okay, through our proteins based on the information on the genome or the DNA that you are inherited from your two parents. Okay, so yeah, now we got it. We have all the information in the whole DNA, but I told you, and this is true, I mean, it could be a little bit more over 3%. What do you think the function or the reason that we have all this DNA? Why do we have it all if we are using only 3% for protein synthesis? What do you think? You don't have to know it, but just try. Did you ask yourself before, why do we need this DNA if we are not using it all? Zainab, can you try? I don't want you to put you on the hot spot. Can you try? Okay, can somebody try instead of Zainab? Okay, okay. So so the DNA, I said that these genes could be transcribed into proteins. So the rest of the DNA is that we're trying to produce some other RNAs, like tRNA and ribosomal RNA and mRNA, and the rest could be used for structural purposes, or excuse me. Or maybe they have function that we can discover later on. So I remember when I was in my undergraduates and I was thinking about postgraduates, I learned that to receive your PhD, you have to come up with something unique, okay, something new, okay. So I was thinking, if we have all these experiments and we have all this research, how come I will be able to come up with something novel, something new? And then I went to my graduate school and I saw that while I'm doing my graduate school, every now and then they discover a new protein. Okay, so that means when I started my PhD, by the way, this percentage was only 2% of the genes are typically expressed into protein. Now we know that it's 3%, and the difference is huge. They discovered so many proteins from that time to now. I'm not going to tell you how long was it, but it's a long time. So they discover another 1% of proteins. So yeah, I mean, science is still open, and we can have new information every day about different functions and about even different types of proteins that we can discover. So so there should be a reason. There is nothing just random in our cells. Okay, they are there for a reason, whether we know it or we don't know it. So either that they are directly contain the information in the messenger RNA, or some pieces or sequences can give us different types of RNAs, or they are there just to make the structure of the DNA the correct structure where the enzymes could help. Okay, so I'm going to show you again some other regions that are very essential within the DNA. So this is the idea of the central dogma. Now we have two strands. Okay, this is the template strand, and this is called non-template strand. So the non-template strand will be exactly the same like the transcript, with the exception of the T's are going to be changed into U's. While the template strand is complementary, okay? So whenever you see a G here, it's going to be a C here. If there is an A, it's going to be U. G, C, C, G, T, A, and so on and so forth. So I remember that there is an easy way to remember which one goes with what. So A with a T, apple in a tree, C with a G, car in a garage. So this is how they used to taught us a while ago. So this is a complementary sequences, and they have to bind with each other to bring the correct sequence. What brings the correct sequence? What makes this transcript? What makes this RNA is the RNA polymerase. So we have to have polymerase. Again, polymerase is a protein, okay? So we need to have a template DNA, and this sequence could be different from me and from Dania, okay? But the differences between the human genomes are 99.9% identical, okay? So can you imagine all these human beings on Earth, they have similarity of 99.9% in their DNA. وبعد ده كله يتشاكل ويتعنصر على بعض. So subhanallah. Okay, so now the RNA polymerase will read along this sequence. So that's why those processes are very consistent between organisms because there's high similarity. I mean, 99.9%. This is huge similarities. So the polymerase will look at the sequence here. Number one, it has to unwind over here. So if there's two strands tight, it cannot read. So it unwind, remove them from each other, and bring the nucleotides. And remember that we have a pool of nucleotides hanging in there. And when it's requested, the polymerase now, for example, now this is the transcript. The second one is T. What do you think? Which nucleotide is coming here? The polymerase will continue reading along the template, DNA template. Now it reads T. Which nucleotide should come here?
A.
U, C.
Okay. A, U, C. Why did it not say T here?
Because in RNA there is uracil, not thymine.
Yeah, because we are synthesizing RNA. Okay? Perfect. Thank you so much. Okay, so now we get to this point, and then in the translation lecture, you're going to see how those are changing into a different language, which is amino acids. Okay, so and now this is the transcription processes. We have initiation, we have elongation, we have termination, and those are very consistent between different organisms, even between eukaryotic cells and prokaryotic cells. Okay, so now, as you can see here, we do have our DNA, the template, the transcript, and then finally transcript of RNA, and then finally we have the proteins. Okay, the idea is that the polymerase has to read and bring the correct nucleotide, okay? Sometimes it doesn't bring the correct nucleotide, okay? So that will be reflected into a mutation or abnormalities with the protein products. So if we have here T, G, G, T, T, we expect it to be U, G, G, U, and so on and so forth. But sometimes the polymerase, for some reason, when it reads here T, might bring an A, okay? So that's a mistake. If you read UGG in the genetic code, it will be different from AGG, and it will produce different amino acids that will be part of our protein, and this will mess up sometimes mess up the protein completely. Okay, but I mean, this is abnormalities. The normal thing is that the polymerase will bind to the template, bring the correct nucleotides, and form the RNA. Okay, very important question, and this is a point of confusion. Whenever I ask my students in the exam, most of the time they did not get it correct. So, what will be the product of transcription? In order to do transcription, we have to have DNA template, and we have to have our polymerase, and we have to have nucleotides. If we have all this recipe, what will be the product of transcription? What are we trying to synthesize here?
An RNA strand.
An RNA strand. Which RNA?
Messenger RNA.
mRNA.
Okay, I'm glad that you did not say the same one. So why did you say messenger RNA and the other student said ribosomal RNA? Who's right, who's wrong?
I said mRNA. The same thing.
Both of you said mRNA? Okay. Do we have another answer? Do we just get messenger RNA from transcription?
Yes. Yeah, just messenger RNA.
Okay. So, so that's why I said this is point of confusion. So when you say transcription, transcription is a synthesis of any RNA. If you want to continue to do translation to produce proteins, you will read the information from the messenger RNA, but we need ribosomal RNA and tRNA to help with the process. Okay, but transcription means RNA synthesis, just RNA. You will never see here mRNA or tRNA or rRNA or miRNA. So transcription is a synthesis of RNA. Any type of RNAs will be synthesized by transcription. So polymerase will come, read along the sequence here, bringing the correct nucleotides and produce RNA. Okay. After that, based on the location, based on the sequence on the genome, it will be either messenger RNA, could be tRNA, or could be any form of RNA. Okay, I'm going to ask my question one more time. What is the product of transcription?
RNA synthesis.
Okay, so all types of RNA, please don't forget that, never in your life, because if the product is only messenger RNA, how do you synthesize tRNA? How do you synthesize ribosomal RNA? From where? Okay, so they are produced and synthesized by the same process, but we will use them in a different way, and we are not going to rely on their sequence anymore. We don't need their sequence. We just need the whole RNA that we synthesize. Okay, so now we keep talking about the ribosomal RNA polymerases, RNA polymerases. In prokaryotic cells, I'm going to start with the prokaryotic. By the way, do you need a break or you want me to continue? Hello? Are you good? Do you want me to continue or you need a break?
You can continue.
No preference? Okay. Okay. The polymerases, those are the enzymes that need them to synthesize the RNA. Okay. In prokaryotics, in eukaryotic cells, we have three distinctive polymerases. Polymerase 1, 2, and 3. Each one will have a specific function. But the big picture, polymerases are helping us to synthesize RNAs. Now we need to see the functions of each one of them and how they are doing that. And besides the polymerase, in eukaryotic cells, we have about maybe 50 different proteins that are helping with the process. And of course, we're not going to study the 50 types of proteins. Some of them are very common, some of them are not well studied, but we know that they are involved in the transcription. Okay, so the function of the polymerases. Polymerase 1 will synthesize some types of the ribosomal RNA. In fact, we have 4 types of ribosomal RNAs, 5.8, 18, and 28, and we have the fourth one is 5S. But the 5S is synthesized by polymerase 3 in addition to tRNA. Okay, just forget about the rest. So, function of polymerase 1, synthesis of 3 types of RNAs. Function of polymerase 3, synthesis of tRNA and 5S RNA. Function of polymerase 2 is the synthesis of messenger RNA. So here they wrote it in a different way. It says protein coding genes. Protein codings, those are the messenger RNAs. Okay, because they will result into or be translated into proteins. Okay, so here we have the messenger RNA synthesis, and it will requires, of course, the polymerase 2. And please make sure that you differentiate between the 2 different types of polymerases. So the genome is long, and now the polymerase 2 try to recognize where to find the sequence that's supposed to be transcribed. Okay, of course you have a sequence, and your polymerase has to do something. Polymerase 2, RNA polymerase 2. How does it figure out from where to start? Okay, start what? Start the transcription itself. So there are specific sequences, and this is what I was referring to when I said there are some regions that are not transcribed, but they have some other functions. So these are sequences on the DNA that are very useful and help the polymerase 2 to figure out the starting point of the gene. Okay, I'm going to show you here. So let's assume this is your DNA strand. And of course, all of these are nucleotides all over the place. And this is the starting point. So how did we decide this is the starting point? And how can we convince the polymerase to start from here? Why doesn't it start from here? Why doesn't it start from here? Because in order for the polymerase and the transcription factors to determine the starting point, there are some sequences found upstream of the starting point. Upstream meaning what? Means before the starting point. Okay, now we are telling you this is the starting point. But how did we know? Because when we look at the product, we see that it's starting from here, and it brought the complementary nucleotides from here. But how the polymerase and the transcription factors bound to this region? How did they recognize it? Because we have a very important and characteristic nucleotides are found in here. So about 20 bases away from the starting point, we have what's called data box. Data box are thymine, adenine, thymine, adenine. Before that, okay, we have GC rich region. That means bunch of GC, 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 GC. Okay. Now we get to about 110 nucleotides away from the starting sequence. It's called CAT box, CAAT sequence. Okay. So these are the sequences on the template DNA. Okay. Once we have them, then this will the polymerase will recognize the starting point. Okay, so if this data box is here, then that means this is not the starting point of our expressed or protein or the gene that it's going to be expressed. So this is one of the functions of the functions of the part of the DNA that might not be expressed into a protein. Okay? Okay, so now we know that the polymerase and transcription factors find out about the starting point, then it will do the transcription. Okay, so I just want to draw your attention to the minimum things that we need to have in order to do the transcription. Okay, so the transcription cannot be started without what's called basal transcription complex. Okay, so what do we mean by basal transcription complex? Basal transcription complex divided into two: basal transcription elements and basal transcription factors. Elements are the sequences that I just showed you. Whenever we say elements, that means those sequences on the DNA. When we talk about factors, those are the protein factors that will bind to the sequences upstream, and we call the sequences here, or the region here, we call it the promoter region. The promoter region contains all these sequences. Transcription factors will recognize them, they bound, and then they recruit the polymerase and tell the polymerase, hey, please come and bind here because this is the starting point. So we need the basal transcription complex. That means we need the sequence on DNA and we need the transcription factors. Those are the sequences on the promoter region, which are called elements: data box, CAD, GC rich, and the transcription factors. This is what we say that we have about 50 and more proteins that are involved and binding in a sequential manner. So one will bind, recruit the second one, and so on and so forth, until they all bound in there. They will recruit the polymerase, and some of them even bound after the polymerase. So they are regulatory factors that are very essential for the transcription to happen. Some other sequences like downstream or within the gene itself, also they will regulate the transcription, and we have some molecules that could bind, also called enhancers and silencers. So they either stimulate the function of gene expression or they will inhibit it. It depends on the type of those enhancers and silencers. And most of the time they are also proteins, if not all times. Okay, now we know that in order to make this transcription, we need sequence on the DNA or the elements, and we need to have the polymerase, we have to have some transcription factors, and also we need to have the nucleotides. Once we get to that point, that means we should be able to synthesize our RNA. We started with the messenger RNA, ribosomal RNA, tRNA will be synthesized the same way. And after we make them, now we want to make them a functioning molecules. So messenger RNA, and remember, we're still talking about eukaryotic cells, okay? Those are within the eukaryotic cells. Those modifications do not occur in prokaryotic cells. So in messenger RNA, we have three processes where we add a cap, and always cap for protection, a tail, and splicing that we need to remove the introns. Okay, I'm going to start with the cap. The cap basically is a nucleotide also. Okay, so it's 7-methylguanosine nucleotide that will bind to the 5' end of the messenger RNA, the first nucleotide, okay, and protect the messenger RNA until it will go to translation and the protein is formed, and then after that we will see that the messenger RNA will be degraded. But this is for protection. Poly-A tail, this is a sequence of adenine nucleotides, okay? And look, whenever we are trying to do modifications here for RNA, we just keep adding nucleotides, okay? So adenine nucleotides, they could be short as 20 nucleotides or up to 200 nucleotides, and this will loop when we do the translation and help the proteins that are needed for translation to bound to the correct sequence. Splicing, when we transcribe an RNA, we always just copy the nucleotides from the DNA. Not all of them are needed for the final product of RNA. Just like the DNA sequence, we just talked about it and we said that not all nucleotides are needed for the function of the DNA. Now, in the RNA also, we bring those sequences with us, but we don't need them. So we have a process called splicing, and the machine here that helps us is called spliceosomes. And what do? They cut the introns out and ligate the exons, and finally the mature messenger RNA should have this structure without those introns. So what do we do with those introns? Introns are nucleotides. We just remove them into the circulation, and we can use them again to synthesize any of those RNAs. So by doing this, our messenger RNA should be ready in this structure, as you can see it here. If we start with introns and exons, without a cap, without a tail, now we are going to do all this, removing introns. Now we have just exons here, building a cap and making a poly-A tail. Once we get to this point, our RNA, our messenger RNA, is ready to go. For ribosomal RNA, also, we are synthesizing it as a transcript, a long one, and then after that, we just cleaved it into different ribosomal RNAs, but they don't have caps, they don't have tails, just different types of RNAs, and we will wait for polymerase 3 to synthesize the last one, and they all together they combine to form our ribosome. And by the way, ribosome here has a catalytic activity called ribozyme. So all our enzymes are proteins, but we have a couple of exceptions. This is one of them. Ribozyme are having the catalytic activity. Okay, tRNA, the last one here. tRNA, I already talked about modifications within the nucleotides. So we synthesize it as a long one. So if you stretch the two ends, just imagine that you are stretching, pull from here and pull from here. It's going to be a long transcript. Then it will take the clover leaf structure, and after that, some of the nucleotides are removed, some of them are modified, and this is the final structure of the tRNA. How we are going to use it? We are going to use it in the translation because it has a specific sequence over here, CCA. This is where an amino acid will bind to it. Okay, so this is the final structure of the tRNA and ready to enter translation. Of course, we are not going to read these sequences to bring amino acids because the only sequence that we are reading are embedded within the messenger RNA. Okay, if we understand the transcription for eukaryotic cells, I'm going to just show you the differences here because the process is always the same. So now the question, what's the function? How do you define transcription in prokaryotic cells? Transcription in the prokaryotic cells. What will be the products of transcription in prokaryotic cells? Is it messenger RNA? Ribosomal RNA? Okay, let's try one more time. tRNA or all of them? Just make an educated guess.
messenger RNA I don't think that they have ribosomal RNA in the prokaryotic cells.
Okay, good try. Okay, at least you got one. You said messenger RNA. That's correct. Any other RNAs? Okay, so we do have tRNA and ribosomal RNAs in prokaryotic cells. Okay, so we are looking at the same products, but it's going to be a little bit different because in eukaryotic cells, the RNA will be synthesized within the nucleus. Okay, so this is the location. You know that your DNA is found in the nucleus. So DNA will be transcribed to messenger RNA. Once we have a mature messenger RNA, then it will leave the nucleus through certain pores. هذه فتحات صغيرة كده هنا بيطلع بيها المسنجر RNA عشان يمشي للريبوزوم where the protein will be synthesized. In prokaryotic cell, this process is a little bit different because prokaryotes do not have the nuclear membrane. So they have the DNA found here in the cytosol, transcribe it to messenger RNA, and then finally we do translation and get our protein in the same region, okay? Cytosol. Here we'll differentiate, okay? We have to start in the nucleus and then we go to ribosome. Okay, so prokaryotic cells also need a DNA to read through it, and also they need polymerase, but here the polymerase is different. It has 4 subunits. In some textbooks, sometimes they say 5 subunits, but I think it's better to say 4 subunits, and there is a initiatory factor which is called sigma factor. So this is a protein that bound to the enzyme at the beginning, just during initiation. Okay, and then we should be able to start the transcription or the initiation process by allowing that polymerase to bind to the promoter region. Again, what is the promoter region? Are you all understand this term, promoter region? Okay, sequence of nucleotides upstream of the starting point. Okay, upstream means before the starting point. So here, if this is my starting point, then I'm going to see the promoter region here, and this is where the polymerase will bind. Okay, again, just like eukaryotic cells, prokaryotes should have certain sequences. They are more simpler, like they have Tata box and the sequence TTGACAT, around 10 nucleotides before the starting point, and this one around 35 nucleotides before the starting point. And when we have these sequences, the polymerase will bound and start the process. So for the initiation, the polymerase should bind to the sigma factor. Can you see this small yellow ball here? This is sigma factor bound to the blue polymerase, start transcription, and once we get a small sequence, now sigma factor is not needed anymore. It helps the polymerase to bind to the correct location, but after that we don't need it, so it will be removed. The transcription will continue until getting to a certain point over here. So take home message from prokaryotic cells that they need sigma factor for initiation. RNA polymerase is the one that unwinds the DNA so it can read through it, and it will bring the correct nucleotides, and always the first one is always a purine. And also the sigma factor will dissociate after it starts the transcription and building or bringing several nucleotides before it dissociates. So elongation is very simple. Just keep, keep unwinding and bringing nucleotides, moving along the fork here and unwinding, removing them from each other, bringing the correct nucleotide until it gets to the final point, which is the termination. Termination in prokaryotic cells could be done by two different mechanisms, either by having a factor, protein factor, and this is very common. Normally when we do any process, we are trying always to find the protein factor, or by a mechanical activity right here. So the RNA will form a loop when it gets to the final area or final point. It makes a loop. So this is the RNA itself, all of it, okay? And loop that has base pairing. So can you see here, U with A, C with G, G with C, and the formation of this loop will tell the polymerase to dissociate from the DNA. It will be removed. If it's removed, the process will stop. So this is one of the mechanisms. The second one requires a protein factor called Rho factor. So when Rho factor binds, the polymerase cannot bind to the DNA and it will dissociate, and the factor will destabilize the polymerase template interaction. So there should be an interaction between the polymerase and the DNA template, and this factor will destabilize it and prevent the binding, so the polymerase cannot read anymore through the DNA and it will stop the transcription. Okay, so the $1 million question: who brought this Rho factor, or who tells the RNA to form this loop? Because these functions, our cellular functions, are regulated mostly by hormones, so those are hormonal signals. If we don't need more proteins, if we don't need those RNA polymerases, we should see that their hormonal involvement, sending signals like, recruiting this protein factor or making this loop over here and inhibit the transcription. Okay, the modifications that's happening within the RNAs are limited, but we have a very interesting process here for messenger RNA in prokaryotic cells. The messenger RNA in eukaryotic cells has separate genes. Each one of them has different promoter region. So each one of them will be expressed differently, okay? So the gene for eye color has its own promoter region. It will be expressed and give the color of the eye, whether it's brown or blue or gray, okay? And the gene for the color of the skin also will be expressed differently. In prokaryotic cells, there are set of genes that are expressed together, whether they are initiate the expression or inhibited together, okay? So whether they are stimulated or inhibited, okay? So this is essential property for prokaryotic cells because it helps them to replicate very fast, okay? And the genes that are expressed together or be expressed into proteins together, this is called a polycistronic transcript. So let's assume that there are 3 genes in a row. They will be regulated by the same promoter region expressed together. The transcript is called polycistronic transcript. And when it translated into proteins, the 3 genes will give 3 proteins together. So those 3 proteins are regulated together and produced also together in the polycistronic transcript. We don't have this property in our eukaryotic cells. Okay, ribosomal RNA and tRNA are produced the same way and also as long transcript, and then you know that it will be divided, okay, or cleaved, and tRNA will also be large and then cleaved to get a smaller one. Okay, so the most modifications that are happening within our eukaryotic cells, but I have to tell you a little bit about those prokaryotic cells because we don't survive without those prokaryotes. They are there in our systems either as uh, harmless bacteria, for example. We have them in our guts, so we need to learn about their mechanisms. And also, they are pathogens that affecting our cellular activities and our health on daily basis. So we need to learn about them. And if we learned about them, then we know the mechanisms by which we can regulate them. So if you have certain types of bacteria and you know that they are replicating using certain enzymes or factors, then you can design a drug that will inhibit that enzyme, and the enzyme will be inhibited and the process will be inhibited. Okay, well, this will end my presentation for today, and I can take any questions. Any questions, please? No questions? Okay, perfect. Let me try to stop recording.